Chapter 16: Disease and Epidemiology. In developed nations like United States, public health is a critical responsibility of the government, as a healthy population contributes to greater productivity and economic prosperity. Conversely, high rates of illness and death can hinder economic growth and lead to social and political instability. When citizens struggle with disease, their ability to work consistently, maintain employment, and build wealth is significantly impacted, affecting not only their lives but also the broader economy. This chapter delves into the relationship between microbiology and epidemiology, the foundational science of public health. Epidemiology focuses on the understanding the origins and spread of diseases within populations, aiming to prevent outbreaks and manage those that occur. Over the past 2 centuries, advancement in epidemiology have led to transformative public health policies that have eradicated or nearly eradicated many diseases that previously caused immense suffering. Nonetheless, the challenges remain, as numerous diseases continue to affect populations, new diseases are emerging, and inadequate infrastructure in developing nations complicate efforts to control disease spread. The learning objective of Section 16.1 include explaining the difference between the prevalence and incidence of disease, distinguishing the characteristic of sporadic, endemic, epidemic, and pandemic diseases, and explaining the use of Koch's postulates and their modifications to determine the etiology of the disease. Additionally, the objectives involved elucidating the relationship between epidemiology and public health. The field of epidemiology focuses on the geographical distribution and timing of infectious disease occurrences, as well as their transmission and maintenance in nature, with the primary aim of recognizing and controlling outbreaks. This scientific discipline encompasses the study of etiology, which investigates the cause of disease, and the exploration of disease transmission mechanisms that detail how diseases spread. Epidemiology is the study of how diseases affect the health and illness of populations. It serves as a cornerstone for public health, providing critical insights into the patterns, causes, and effects of health conditions in specific populations. By employing statistical method and rigorous scientific research, epidemiologists gather, analyze data, identify risk factors, and trends associated with diseases. One key aspect of epidemiology is its focus on evidence-based practices. This approach emphasizes the importance of using empirical data to inform public health policies and interventions. Epidemiologists utilize studies such as cohort studies, case-control studies, and randomized controlled trials to establish causal relationships and evaluate the effectiveness of treatment and preventive measures. For instance, during the COVID-19 pandemic, epidemiologists played a vital role in tracking the virus's spread, identifying high-risk populations, and assessing the impact of vaccination campaigns. Their findings informed government responses and public health guidelines, ultimately saving lives. In summary, epidemiology is an essential science, but not only enhances our understanding of the disease, but also shapes health policies through evidence-based research. Its contributions are crucial for effective disease prevention and health promotion in diverse communities worldwide. From 1900 to 2020, the landscape of mortality in the United States underwent significant transformations, reflecting changes in lifestyle, medical advancement, and public health interventions. In the early 20th century, infectious diseases such as pneumonia, tuberculosis, and gastrointestinal infections dominated the list of leading causes of death. However, as public health measures improved, such as sanitation, vaccination, and antibiotics, the mortality burden shifted. By the mid-20th century, chronic diseases began to emerge as primary causes of death. Conditions such as heart disease, cancer, and stroke became more prevalent, largely due to the lifestyle factors like smoking, poor diet, and sedentary behavior. Epidemiological studies highlighted the role of these factors in the development of chronic illnesses, leading to targeted public health campaigns aimed at prevention. By 2020, the COVID-19 pandemic introduced a new challenge with respiratory diseases once again rising to the forefront of mortality statistics. This shift underscored the dynamic nature of epidemiology, demonstrating how emerging threats can reshape health priorities. However, the evolution of causes of death in the USA reflects the complex interplay of social, environmental, and biological factors that influence population health over time. Epidemiological analyses are always conducted with reference to a population, which consists of individuals at risk for a specific disease or condition. This population can be defined geographically. However, if only a portion of the individuals in that area are at risk, additional criteria may be necessary. Susceptible individuals can be identified based on their certain behaviors, such as intravenous drug use, owning specific pets, or being part of an institution like a college. Defining the population is crucial because most measures of interest in epidemiology are calculated with respect to the population size. There are two aspects of morbidity that are relevant to the epidemiologist: a disease's prevalence and its incidence. Prevalence is the number or proportion of individuals with a particular illness in a given population at a point in time. For example, the WHO estimates that 39.9 million people were living with HIV at the end of 2023. Approximately 1.3 million new HIV infections were reported in 2023. HIV-related deaths: around 630,000 people died from HIV-related causes in 2023. While there have been notable declines in new infections and deaths since the peak of epidemic, HIV remains a significant global health challenge, particularly in sub-Saharan Africa. Approximately 86% of the people living with HIV globally knew their HIV status in 2023. But about 5.4 million remains unaware. In 2023, 30.7 million people were assessing antiretroviral therapy. For a chronic disease like HIV infection, prevalence would generally be higher than incidence because it represents a cumulative number of new cases over many years minus the number of cases that are no longer active, example, because the patient died or was cured. Measuring infectious disease frequency is essential for understanding the spread and impact of diseases within populations. Key matrices include incidence and prevalence rates. Incidence measures the number of new cases in a specified time frame, indicating the risk of infection. Prevalence assesses the total existing cases at a particular moment, reflecting the disease's burden on healthcare systems. Other important factors include morbidity rate, which indicates the proportion of a population that becomes ill during an outbreak, and mortality rate, which measures the severity of the disease by calculating the percentage of deaths among confirmed cases. These matrices guide public health responses. Morbidity rate and mortality rate are critical epidemiological indicators used to quantify health outcomes in populations. Morbidity rate refers to the prevalence of a disease or health condition within a specific population during a certain time frame. It encompasses both the incidence, new cases, and the existing cases of a disease. Morbidity rates are often expressed as the number of cases per 1,000 or 100,000 individuals. This metric helps public health officials understand the burden of the diseases, assesses the effectiveness of health interventions, and allocate resources for healthcare services. Mortality rate, on the other hand, measures the frequency of deaths in a given population over a specific period, typically expressed as the number of deaths per 1,000 or 100,000 individuals. This rate is crucial for evaluating the overall health of a population and the impact of various diseases. Mortality rates can be categorized into specific types, such as infant mortality rate, deaths of infants under 1 year of age, and cause-specific mortality rate, deaths due to a particular cause. Together, morbidity and mortality rates provide valuable insight into public health, enabling policymakers to implement effective strategies to improve population health outcomes. Incidence rate and prevalence rate are fundamental concepts in disease epidemiology that help researchers and public health officials understand the dynamics of health conditions within populations. Incidence rate refers to the number of new cases of a disease that occur in a specified population during a defined time period. It is usually expressed as the number of new cases per 1,000 and 100,000 individuals at risk. Incidence rates are crucial for identifying the risk of developing a disease and are instrumental in studying the causes and patterns of disease over time. Prevalence rate, on the other hand, measures the total number of existing cases, both new and pre-existing, of a disease within a population at a specific point in time or over a specified period. This rate is typically expressed as the number of cases per 1,000 or 100,000 individuals. Prevalence provides insights into the overall burden of a disease in a community, helping to inform healthcare planning and resource allocation. Sporadic diseases differs to the disease that occurs infrequently and irregularly in a population. These cases are isolated and not part of a broader trend or pattern. Sporadic diseases may arise due to unique circumstances, such as individual exposure to a pathogen or environmental factors. Sporadic diseases are those that occur infrequently and without a specific geographic concentration, with example includes tetanus, rabies, and plague. In the US, the bacterium Clostridium tetani, which causes tetanus, is commonly found in soil, yet infections are rare due to the widespread vaccination, proper wound care, and limited exposure to infection risks. Similarly, plague cases are infrequently reported each year, typically arising from contact with rodents in rural western states. Endemic diseases are constantly present, often at a low level in a population within a particular geographic region. The occurrence of endemic disease is relatively stable over time, with predictable rates of infection. These diseases are often maintained through ongoing transmission among the population. A classic example of an endemic disease is malaria in certain tropical regions and some regions of Brazil, but is not endemic to the United States. Hyperendemic disease refers to a situation where the prevalence of a disease is consistently higher than what is typically expected in a particular area or population over a prolonged period. Unlike an epidemic, which denotes a sudden increase in cases, hyperendemic status reflects a sustained high level of disease activity. An example might be a region experiencing high rates of tuberculosis consistently over years. Outbreak describes a sudden, unexpected occurrence of a disease within a specific population or geographic area. Outbreaks can involve a small number of cases and are often limited to a particular time frame. They may be triggered by various factors such as environmental changes or a new pathogen introduction. Epidemic is defined as a significant increase in the number of cases of a disease within a population, affecting many people simultaneously. Epidemics can occur when a new strain of a pathogen emerges or when existing pathogens spread more rapidly than usual. They often require public health interventions to control the spread. Influenza is a good example of a commonly epidemic disease. Pandemic signifies an increase in disease occurrence that affects a large population across multiple countries or continents. A pandemic usually involves a new infectious disease that spread easily from person to person, resulting in widespread health impacts. An example is COVID-19, which originated in China and rapidly spread across the globe, impacting millions of people. An epidemic disease signals the breakdown of an equilibrium disease frequency, often resulting from the changes in the environmental conditions or in the population. In the cases of influenza, the disruption can be due to antigenic shift or drift, which allows influenza virus strains to circumvent the acquired immunity of their human hosts. Incidence patterns of influenza tend to rise each winter in the northern hemisphere. These seasonal increases are expected, so it would not be accurate to say that influenza is epidemic every winter. However, some winters have an unusually large number of seasonal influenza cases in particular regions, and such situations would qualify as epidemics as shown in the slide figure. The seasonal epidemic or threshold represented by the blue curve on the graph is established using data from the Center for Disease Control and Prevention (CDC) from 2004 to 2008. This threshold serves as a critical benchmark for monitoring the health of the population, specifically regarding mortality rates associated with pneumonia and influenza. When actual mortality rates surpass the predefined threshold, it indicates a significant increase in disease prevalence, leading to classification of the situation as an epidemic. The graph illustrates distinct spikes in pneumonia and influenza-related mortality during several winter seasons, particularly in 2003, 2004, 2005, and 2008. These pronounced epidemics highlight the seasonal nature of respiratory illnesses, emphasizing how they often peak during colder months when individuals are more likely to be close contact with one another, facilitating virus transmission. Additionally, factors such as fluctuations in temperature, humidity, and population behavior during the winter months can contribute to increased vulnerability to these diseases. Understanding these patterns is vital for public health officials and healthcare providers, as it informs preparedness and response strategies, including vaccination campaigns and resource allocation. By analyzing historical data and trends, health authorities can better anticipate and mitigate the impact of future epidemics, ultimately striving to protect the health and well-being of the community. When studying an epidemic, an epidemiologist's primary task is to identify the cause of the disease, known as the etiologic or causative agent. Establishing a direct connection between disease and a specific pathogen can be challenging, as it requires controlled experiments to rule out other possible causes. This difficulty is compounded by the nonspecific nature of disease signs and symptoms, which can be caused by multiple agents. As a result, diagnosing a causative agent often proves complex, particularly where there are no clear indicators of the outbreak's cause. Historically, Robert Koch was the first scientist to demonstrate a direct causative agent of a disease, specifically anthrax, in the late 1800s. He established 4 criteria Koch postulates that must be met to link a disease with a microbial pathogen, paving the way for the golden age of microbiology. Between the 1876 and 1905, several common diseases such as cholera and tuberculosis were linked to their etiologic agents. Today, molecular Koch postulates, a modern adaptation of the original criteria, are utilized to connect disease states with specific virulence traits of pathogenic strains, furthering our understanding of how pathogens cause disease. In contrast, complex diseases often present as a diagnosis riddle, further complicated by combination of multiple phenotypes and disease as feature of these diseases. With the aim of enhancing the determination of key etiological factors, we developed and tested a complex disease model that encompasses diverse factors that in combination result in complex diseases. This model was developed to address the challenges of classifying complex diseases given the evolving nature of understanding of disease and interactions and contributions of genetic, environmental, and social factors. Epidemiology began with physicians who analyzed disease patterns to understand prevention. Girolamo Francescaro proposed the germ theory of disease in 1546. But it competed with miasma hypothesis for years. Despite uncertainty about disease causes, researchers like Florence Nightingale utilized existing knowledge. The 19th century saw a shift towards the germ theory influenced by the key figures such as John Snow, Nightingale, Ignaz Semmelweis, Joseph Lister, Robert Koch, and Louis Pasteur, laying the foundation for modern epidemiology. John Snow, a British physician known as the father of epidemiology, played a pivotal role in understanding the 1854 Broad Street cholera epidemic in London. His earlier observation during 1848-1849 cholera outbreak led him to propose that cholera was spread via a fecal-oral route and that a microbe was the infectious agent. Investigating the 1854 epidemic, Snow focused on the water source of the infected individuals. He discovered a high incidence of cholera among those using water from River Thames, contaminated with refuse and sewage from upstream settlements, and noted that brewery workers who consumed beer instead of water did not contract cholera. Snow's efforts represented an early epidemiological study and initiated the first known public health response to an epidemic. His meticulous case of tracking methods are now standard in disease outbreak investigations, helping to link new diseases to their causes. His work also highlighted the unsanitary practices related to the sewage and detrimental effects of waste dumping into the Thames. Moreover, Snow's findings supported the germ theory of disease, which posits that disease could be transmitted through contaminated items such as fecal matter in water. Snow identified what is now termed as common source spread of infectious disease, where all infected individuals share a single source. In the case of Broad Street cholera outbreak, the contaminated well below the pump was identified as the source. Common source spread can take various forms, including point source spread, where infections occur from a short period, continuous common spread lasting longer than the pathogen's incubation period, and intermittent common source spread, where infections occur in cycles. An example of point source spread would be the single contaminated dish at a picnic, while continuous common source spread is exemplified by contaminated water from London. Florence Nightingale's contribution to the early epidemiology emerged during her service in the Crimean War in 1854, where she had a group of nurses were tasked with caring for wounded soldiers. Throughout the period, Nightingale meticulously recorded the causes of illness and death, a foundational aspect of what would later evolve into the science of epidemiology. Her findings were published in 1858, showcasing her analysis through innovative graphical methods such as a wave chart histogram that displayed monthly frequency data on mortality causes. Nightingale's data presentation was groundbreaking for its time and compellingly demonstrated that most casualties were not the result of battle injuries, but rather preventable infectious diseases, often linked to poor sanitation and inadequate hospital facilities. Her work highlighted the urgent need for reforms in medical care within the British military, ultimately leading to significant improvements in health practices and sanitation standards. Joseph Lister played a crucial role in advancing public health practices in clinics and hospitals during the mid-1800s, a time when surgical wound infections were common and the germ theory of disease was not widely recognized. Many physicians neglected basic hygiene, failing to wash hands or sanitize surgical tools, which contributed to high rate of post-surgical infections. Lister's groundbreaking discovery of the disinfecting properties of carbolic acid or phenol led him to implement effective disinfection protocols, significantly reducing infection rates. He mandated the use of 5% carbolic acid solution for cleaning the surgical instrument and even applied it to the bandages and surgical sites during operation. In addition to these measures, Lister took personal precautions to prevent introducing infections from his own body or clothing. He would remove his coat, roll up the sleeves, and wash his hands with dilute carbolic acid solutions before and throughout surgeries. Lister's innovative practices laid the groundwork for modern antiseptic techniques, demonstrating the importance of cleanliness and disinfection in medical settings. His contributions were pivotal in improving patient outcomes and transforming surgical procedures. Common source outbreaks occur when a group of people are exposed to the same source of infection, such as contaminated food or water. There are different types within the category, including point source, continuous source, and intermittent source outbreaks. Point source outbreaks involve a brief exposure to a single source, with cases arising within one incubation period. For example, a food poisoning outbreak from a contaminated buffet would be point source outbreak. Continuous source outbreak involve ongoing exposure to the same source over a period of time, leading to a gradual increase in cases. A cholera outbreak from a contaminated water well would be a continuous source outbreak. Intermittent source outbreaks involves exposure to the same source at irregular intervals. This might happen if a restaurant fails to fix a refrigerator and has multiple outbreaks of food poisoning. Propagated outbreaks occur when the infection spreads from person to person either directly or indirectly. Common source outbreaks involve exposure to a single shared sources like contaminated food or water. Mixed epidemics combine aspects of both, starting with a common source and then spreading through person-to-person contact. Some other type of outbreaks can be classified as a point source, continuous source, and intermittent source outbreaks and shows a distinct individual pattern of its own. All of the mentioned patterns of epidemics will be discussed in the subsequent slides. A common source outbreak is the one in which a group of persons are all exposed to an infectious agent or a toxin from the same source. If the group is exposed over a relatively brief period, so that everyone who becomes ill does so within one incubation period, then the common source outbreak is further classified as a point source outbreak. The epidemic of leukemia cases in Hiroshima following the atomic bomb blast and the epidemic of hepatitis A among the patrons of Pennsylvania restaurant who ate green onions, which had a point source of exposure. If the number of cases during an epidemic were plotted over time, the resulting graph, called an epidemic curve, would typically have a steep upslope and a more gradual downslope, is so-called a log normal distribution. In some common source outbreaks, case patients may have been exposed over a period of days, weeks, or longer. In a continuous common source outbreak, the range of exposures and the range of incubation periods tend to flatten and widen the peak of the epidemic curve as shown above. The epidemic curve of an intermediate point source outbreak often has a pattern reflecting the intermediate nature of exposure. A propagated outbreak results from the transmission from one person to another. Usually transmission is by direct person-to-person contact, as with syphilis. Transmission may also be vehicle-borne, transmission by hepatitis B or HIV by sharing needles, or vector-borne, transmission of yellow fever by mosquitoes. In propagated outbreaks, cases occur over more than one incubation period. In this slide, note the peak occurring about 11 days apart, consistent with the incubation period for measles. The epidemic usually wanes off after a few generations, either because the number of susceptible persons falls below some critical level required to sustain transmission, or because intervention measures become effective. Some epidemics have features of both common source epidemics and propagated epidemics. The pattern of a common source outbreak followed by a secondary person-to-person spread is not uncommon. These are called mixed epidemics. For example, a common source epidemic of shigellosis occurred among a group of 3,000 women attending a national music festival, many developing symptoms after returning home. Over the next few weeks, several state health departments detected subsequent generations of Shigella cases propagated by person-to-person transmission from festival attendees. Finally, some epidemics are neither common source in its usual sense nor propagated from person to person. Outbreaks of zoonotic or vector-borne diseases may result from sufficient prevalence of infection in host species, sufficient presence of vectors, and sufficient human-vector interaction. Examples in this slide figure include the epidemic of the Lyme disease that emerged in the northeastern United States in the late 1980s, spread from deer to human by deer ticks. The A histogram shows a general increase in the trend of the number of reported cases of the Lyme disease. The B histogram shows the reported cases of West Nile encephalitis in the Queens section of New York City in 1999 and other locations, spread from birds to human by mosquitoes. Cases dropped to zero after the mosquito control activities began in the city. Reported cases in other locations continue at about the same rate. Epidemics are complex phenomena characterized by the rapid spread of infectious disease within populations. The propagation of epidemic is largely influenced by the concept of threshold density, which refers to the minimum number of susceptible individuals required within a population to sustain an outbreak. When the density of susceptible host reaches this threshold, the likelihood of the disease transmission increases, allowing epidemic to flourish. Susceptible individuals are those who have not been infected or immunized against the disease in question. Their presence is crucial for the spread of the epidemic, as they provide necessary host for the pathogen to replicate and disseminate. The dynamics of the outbreak can be modeled using mathematical frameworks that incorporate factors such as transmission rate, recovery rate, and the number of susceptible individuals. As cases arise, the epidemic can either grow or decline based on the interaction among individuals. If the number of new infections surpasses the recovery rate, the epidemic will persist and potentially escalate. Conversely, if the number of susceptible dwindles, either through infection or vaccination, the epidemic may eventually subside. Understanding these dynamics is essential for public health interventions aimed at controlling and preventing the spread of infectious disease. The slide graph shows a number of individuals on the vertical axis and the time of days on the horizontal axis. The vertical axis is unmarked. The horizontal axis ranges from 0 through 30 with intervals of 5. The introduction of an infected individual is at the origin. The lower curve is bell-shaped with a peak of about 15 days representing the number of cases. The upper curve, susceptible starts at the maximum initially, form an S-curve at the end at 30, the same level as the maximum of the curve of the case. The peak of the epidemic wave coincides with the threshold density of susceptible individuals. Observational studies are research methods where data is collected from participants through various means such as measuring physiological variables or conducting interviews about lifestyle factors. Participants are typically selected randomly from either affected or unaffected populations. Unlike experimental studies, researchers do not manipulate the subjects in these studies. Observational studies are often easier to conduct and may sometimes be only feasible options due to ethical considerations. These studies primarily measure associations between disease occurrences and potential causes without establishing causation. For example, an association found between heavy coffee consumption and reduced skin cancer rates does not prove that coffee prevents cancer. Other factors, such as sun exposure, could be influencing the results, demonstrating the limitations of observational studies in distinguishing between different potential causes. There are two primary approaches in observational studies: descriptive epidemiology and analytical epidemiology. Descriptive epidemiology focuses on gathering information about disease outbreaks, affected individuals, and the disease's spread during an exploratory phase. This may involve interviews, medical record and examination, and food histories, often while the outbreak is ongoing. Such studies can aid in forming hypotheses for further testing in more rigorous experimental studies. Analytical epidemiology, on the other hand, involves selecting specific groups of individuals to evaluate hypotheses about diseases cause more convincingly. Random selection helps avoid bias related to shared characteristic among participants. Analytical studies can be retrospective, looking back in the time, or prospective, following events as they occur. Retrospective studies analyze the past data related to current cases, considering factors like medical history, demographics. These studies help identify associations between various factors and disease occurrences, providing insight into potential causes. Prospective studies monitor individuals over time, collecting data on their characteristics and environments. By comparing those who develop a disease With those who do not, researchers can identify associations between disease states and measured variables, contributing to understanding possible cases. Analytical studies also utilize group-based approaches, including cohort studies, case-control studies, and cross-sectional studies. Cohort studies examine group sharing specific characteristics, such as birth year or behaviors like smokers versus non-smokers. These studies can be conducted either retrospectively or prospectively, allowing research to analyze disease incidents correlated with their certain characteristic without ethical violations. Case-control studies are primarily retrospective, comparing individuals with a disease to similar individuals without a disease. This method is more efficient than cohort studies, as researchers can select already affected subjects instead of waiting for disease to develop in a random sample. Cross-sectional studies involve analyzing randomly selected individuals at a specific point in time, comparing those with a disease to those without. These studies help identify associations between measurable variables and diseases and are also helpful for determining the prevalence of conditions within a population. Experimental epidemiology involves laboratory or clinical studies where researchers manipulate study subjects to explore links between disease and potential causative agents or to evaluate treatments. These treatments can include administering drugs, altering diets, promoting physical exercise, or performing surgical procedures. Both animals and humans can serve as test subjects in these experiments. However, due to the ethical implication of manipulating subjects, conducting experimental studies can be challenging and in some cases impossible. To identify the causative agents of diseases, Koch's postulate necessitates experimental interventions. Unlike observational studies, experimental studies yield stronger evidence of causality since researchers can control other variables while manipulating subjects. In these studies, outcomes from a treatment group receiving an intervention, such as a specific medication, are compared to a control group that does not receive the treatment but is otherwise treated identically. This setup allows researchers to isolate the effects of treatment by ensuring that any changes observed are attributable to the specific manipulation. While experimental studies offer the most robust evidence regarding disease etiology, they must be meticulously designed to mitigate bias. A common approach in human experimental studies is the double-blind design, where neither the participants nor the researchers know who is receiving the treatment and who is not. This method effectively reduced the placebo effect and known bias, where the awareness of the treatment can inadvertently influence the study's outcome. By employing such designs, researchers enhance the reliability of their findings in establishing causal relationships within epidemiology. Understanding the spread of infectious pathogen is essential for disease prevention. While many pathogens depend on a living host for survival, some can remain dormant outside of hosts. Once a pathogen infects a host, it must have a means to transfer to another host to survive, or it risks extinction when its current host dies. Pathogens often develop complex adaptations that take advantages of hosts' biology, behavior, and ecology to facilitate their movement between hosts. In response, hosts have evolved defenses against pathogens. However, due to slower evolutionary rate, hosts generally find themselves at a disadvantage. This section will delve into the environments where pathogens thrive both within and outside the host and examine the various mechanisms by which they transfer from one host to another. Pathogens need reservoir to survive over extended periods, which can be either living organisms or non-living sites. Non-living reservoirs include environments such as soil and water that can naturally harbor pathogens due to their growth conditions. These environments may also become contaminated with pathogens through human feces or the remains of the intermediate hosts. The ability of pathogens to persist in these non-living environments is crucial for their survival and potential spread. Many pathogens possess mechanism that allows them to enter a state of dormancy or resilience enabling them to survive without reproducing in non-living environments for varying lengths of time. For instance, Clostridium tetani can survive in soil as a resident endospore. While most viruses tend to be destroyed quickly upon contact with air or water, some like influenza viruses can survive outside living cells for extended periods. A study revealed that influenza viruses could remain viable on a banknote for 48 hours to 17 days, depending on the conditions, whereas rhinovirus, which causes the common cold, generally survive for less than a day outside physiological fluids. Humans can also act as reservoirs for pathogens, but whether they can transmit the pathogen depends on their infection stage and the specific pathogens involved. To mitigate disease spread among school children, the CDC has established guidelines based on the transmission risk during infection. For example, children with chickenpox are contagious for 5 days from the onset of the rash, while those with the gastrointestinal illnesses should stay home for 24 hours after symptoms have resolved. Carriers are individuals who can transmit pathogen without showing symptoms. A passive carrier who is contaminated but not infected can mechanically transfer the pathogen to another host. For example, a healthcare worker who neglects hand hygiene after interacting with an infected patient can become a passive carrier, potentially transmitting the pathogen to others. This highlights the importance of infection control practices in healthcare settings. In contrast, active carriers are infected individuals capable of spreading the disease. They may exhibit signs or symptoms or remain asymptomatic while still transmitting the infection. Active carriers can transmit pathogen during the incubation period or after symptoms have subsided. Asymptomatic carriers do not display symptoms despite being infected, can transmit disease such as hepatitis B, herpes simplex, and HIV. A well-known example is Mary Malone, or Typhoid Mary, who unknowingly spread typhoid fever while working as a cook, leading to numerous infections and fatalities in New York City. Some pathogens have multiple living reservoirs, particularly in zoonotic diseases where animals serve as reservoirs for human infections. Transmission to humans can occur through the direct or indirect contact with these animal reservoirs. While some zoonotic diseases affect the animal population, others may not show any symptoms in their reservoir host, complicating control efforts. In the context of parasitic infection, the definitive host is the preferred host where the parasite reaches sexual maturity. Complex life cycles may involve one or more intermediate hosts where the parasite undergoes immature life stages or reproduces asexually. Understanding these dynamics is essential for managing and preventing the spread of parasitic disease and ensuring effective treatment protocols. Regardless of the reservoir, transmission must occur for an infection to spread. First, transmission from the reservoir to the individual must occur. Then, the individual must transmit the infectious agent to other susceptible individuals, either directly or indirectly. Pathogenic microorganisms employ diverse transmission mechanisms. Contact transmission. Contact transmission is categorized into direct and indirect contact. Direct contact transmission occurs through person-to-person interactions, where pathogens are transferred via physical contact such as touching, kissing, sexual intercourse, or droplet sprays. This can be further divided into vertical, horizontal, or droplet transmission. Vertical transmission refers to the transfer of pathogens from mother to child during pregnancy, birth, or breastfeeding. Horizontal transfer occurs between individuals through skin or mucous membrane contact, which is often necessary for the pathogen to infect a new host. Certain disease may require specific types of contact or transmission, like sexual contact, while not being transmitted through other contact forms. Droplet transmission, a form of direct contact, happens when an individual coughs or sneezes, releasing small mucus droplets that may contain pathogens. This type of transmission occurs over short distances of 1 meter or less and is responsible for spreading a variety of disease, including influenza and pneumonia. In contrast, airborne transmission refers to the spread of pathogen over distances greater than 1 meter, which can involve different mechanisms for pathogen spread. Indirect contact transmission involves inanimate objects known as fomites that become contaminated with pathogens from an infected person or reservoir. For instance, droplets from a person with a cold can land on surfaces like tablecloths or doorknobs, leading to indirect transmission when a susceptible individual touches these contaminated objects. Fomites can also include improperly sterilized medical equipment such as syringes and catheters, which are significant contributors to healthcare-associated infections. This highlights the importance of hygiene and sterilization in preventing the spread of infections in clinical settings. Vehicle transmission refers to the spread of pathogens through various mediums like water, food, and air. Poor sanitation practices can lead to water contamination, resulting in waterborne diseases, which pose a significant health risk globally. The World Health Organization estimates that contaminated drinking water is linked to over 500,000 deaths annually. Similarly, foodborne diseases arise from improper handling or storage of food, highlighting the critical need for safe water and food practices to prevent disease transmission. Airborne transmission involves pathogens being carried by dust and fine particles known as aerosols. These particles can remain suspended in the air, facilitating the spread of diseases. For instance, hantavirus, found in mouse feces, urine, and saliva, can become airborne when these substances are dry and are disturbed. Inhalation of these particles possesses a severe risk, potentially resulting in fatal respiratory infections. While droplet transmission is categorized as contact transmission over short distances, longer-range droplet transmission is classified as vehicle transmission. Unlike larger droplets that quickly settle, fine mucus droplets from coughs or sneezes can linger in the air for extended periods and travel significant distances. Environmental factors like air temperature and humidity can influence the effectiveness of this airborne transmission, as droplets can quickly dry out, forming droplets, nuclei capable of carrying pathogens. One notable example of airborne transmission is tuberculosis, caused by the bacterium Mycobacterium tuberculosis. When individuals with tuberculosis cough, they release tiny particles that can initiate infection with as few as 10 microbes. Therefore, it is essential for patients with tuberculosis to be treated in specially ventilated rooms, and anyone entering these spaces should wear masks to minimize the risk of transmission. Diseases can be transmitted through mechanical or biological vectors, which are typically animals, often arthropods, that can carry pathogens from one host to another. Mechanical transmission occurs when a mechanical vector transferred a pathogen without being infected itself. An example of this is a fly that lands on a fecal matter and later contaminates food with bacteria from feces. When a human consumes the contaminated food, they become infected, potentially resulting in diarrhea or dysentery. In contrast, biological transmission involves the pathogen reproducing within a biological vector, which then transmit the pathogen to another host. Arthropods are primary vectors for biological transmission, often introducing pathogen through bites that create wounds. For instance, kissing bugs transmit Chagas disease by defecating on the bite site, allowing the infected feces to enter the human body through mucous membranes or skin breaks. This method of transmission highlights the complexity of biological transmission, as the pathogen's life cycle is intertwined with that of its vector. Various arthropods serve as biological vectors, including mosquitoes that transmit malaria and lice responsible for typhus. Other vectors such as ticks and mites can transmit diseases like Lyme disease and scrub typhus, respectively. Additional non-arthropod vectors, including mammals and birds, play a significant role in disease transmission. For example, rabies can be transmitted to humans through bites from infected mammals, while chickens can spread avian influenza through contact with the saliva, mucus, and feces. These diverse transmission mechanisms illustrate the intricate relationship between the pathogen vectors and the host. The slide table shows a few common arthropods vectors and a few selected pathogens. Individuals suspected or known to have been exposed to contagious pathogens may undergo quarantine or isolation to prevent disease transmission. Hospitals and healthcare facilities typically establish special wards to isolate patients diagnosed with hazardous illnesses like tuberculosis or Ebola. These wards are often equipped with advanced air handling systems, and healthcare personnel follow strict protocols to minimize transmission risks. This includes the use of personal protective equipment and chemical disinfectant sprays upon entering or exiting these areas. The length of quarantine is influenced by the disease's incubation period and the presence of infections indicator. Patients may be released from quarantine if they show no signs and symptoms within an expected time frame, or if they can receive preventive treatments to reduce transmission risk. However, if an infection is confirmed, the patient is required to remain in isolation until the disease is no longer contagious, ensuring the safety of public health. In the United States, public health authorities can enforce quarantine measures for specific diseases, including cholera, diphtheria, infectious tuberculosis, and certain influenza strains that could lead to a pandemic. The CDC has the authority to quarantine individuals entering the U.S. or moving between the states if they are suspected of exposure to these diseases. Although the CDC monitors entry points for individuals exhibiting illness, the actual implementation of quarantine is infrequent. Hospitals, retirement homes, and prisons are critical areas of focus for epidemiologists due to their increased incidence of specific diseases. The heightened rates of transmission in these environments can be attributed both the characteristic inherent to the settings and the populations they serve. To mitigate the risk of infection within these facilities, it is essential to implement targeted strategies and interventions. Infections that occur within healthcare settings, including hospitals, are referred to as nosocomial infections or healthcare-associated infections, HAIs. These infections are often linked to surgical procedures or other invasive treatment, allowing pathogens to enter the body. For an infection to be classified as an HAI, the patients must have been admitted for a condition unrelated to the infection itself. Patients in these facilities frequently have compromised immune systems due to their primary illnesses, making them more vulnerable to secondary infections and opportunistic pathogens. According to the CDC, over 720,000 HAIs are reported in the U.S. hospitals in 2011. Surgical sites infections, pneumonia such as accounted for about 22% of the cases, while the urinary tract infection represented 13% and primary bloodstream infections constituted about 10%. These infections often arise when pathogens are introduced through contaminated medical equipment, including catheters and ventilators. To combat nosocomial infections, healthcare facilities implement rigorous training and hygiene protocols aimed at controlling microbial growth. The human parts affected due to infections caused by bacteria, fungi, viruses through factors like healthcare workers and contaminated instruments include ventilator-associated pneumonia in lungs, intestinal tract and liver, urinary tract, bloodstream, and surgical site. The types of microorganisms of the bacteria, fungi, and viruses as shown in the figure. The Centers for Disease Control and Prevention is the primary national public health agency in the United States operating under the Department of Health and Human Services, with the mission of protecting the public from disease and injury. A key component of the mission is National Notifiable Disease Surveillance System, which collaborates with various public health departments to monitor disease of significant public health concern, referred to as notifiable or reportable diseases. Physicians must report cases of these diseases, which include HIV, measles, and West Nile virus, to the CDC. Some states maintain their own list of notifiable diseases. Epidemiological studies track these diseases, and the data is disseminated to healthcare providers and the public through Morbidity and Mortality Weekly Report, which offers updates on public health issues and relevant data. International public health issues are primarily coordinated by the World Health Organization, a United Nations agency with multi-billion-dollar budget. In addition to the member state allocations, more than half of the operating budget comes from voluntary contributions. WHO plays a crucial role in monitoring and reporting on infectious diseases that has developed effective strategies for their control and prevention. Notably, its vaccination program against smallpox, initiated in the mid-1960s, led to the global eradication of the disease by 1980. The organization continues to focus on infectious disease control, particularly in developing nations, with targeted programs for malaria, HIV/AIDS, and tuberculosis, as well as initiatives aimed at reducing illnesses and mortality from violence, accident, and lifestyle-related diseases. Additionally, WHO maintains a global alert and response system that coordinates information from member nations during public health emergencies or epidemics, providing logistical support and managing international responses. The United States contributes to these efforts through the Center for Disease Control and Prevention, which conducts international monitoring and public health initiatives to safeguard U.S. public health in a global interconnected environment. Similarly, the European Union has established a health security committee that monitors disease outbreaks both within its member countries and internationally, working in collaboration with WHO to enhance global health security. Both the World Health Organization and national public health agencies such as Center for Disease Control and Prevention play crucial roles in monitoring and preparing for emerging infectious diseases. An emerging infectious disease is defined as one that is either new to the human population or has exhibited an increase in prevalence within the last 20 years. The classification of a disease as emerging highlights the necessity for allocating resources to comprehend and manage its growing impact, whether it be due to the disease's novelty or changes in conditions that have led to a rise of its frequency. Emerging diseases are a global concern, affecting both developed and developing nations. However, some countries possess better resources to address these health challenges. National and international public health agencies are particularly vigilant in monitoring outbreaks in developing nations, which often lack the healthcare infrastructure and expertise required to effectively manage large-scale outbreaks. Despite assistance from international organizations, many systems in West Africa struggled to adequately identify and treat the sick while controlling the spread of the disease. Emerging diseases can exhibit gradual changes in their frequency over time or can experience sudden surges that lead to epidemics. The significance of remaining vigilant was underscored during the Ebola hemorrhagic fever epidemic in West Africa from 2014 to 2015. Despite health experts being aware of the Ebola virus since the 1970s, the scale of this outbreak was unprecedented. Previous human epidemics had been small and isolated, with gorilla and chimpanzee populations suffering more severely than humans. However, in 2014, the disease's high transmission rate, along with cultural practices concerning the treatment of the deceased and its occurrence in urban settings, facilitated rapid spread, resulting in thousands of deaths. The international public health community mounted a significant emergency response to treat affected individuals and contain the outbreak. In addition to Ebola, there are numerous other diseases that warrant global monitoring. In 2015, the WHO established priorities for several emerging diseases that had a high potential for causing epidemics and were poorly understood, initiating urgent research and development efforts to better understand them. This proactive approach aims to mitigate the risk associated with these diseases before they escalate into larger public health crises. Reemerging infectious diseases are another critical area of concern, as these are diseases that are increasing in frequency after a prior decline. Factors contributing to their reemergence may include changing environmental conditions or the ineffectiveness of previous prevention strategies. Notable examples of reemerging diseases include drug-resistant tuberculosis, bacterial pneumonia, and malaria, The rise of drug-resistant strains of bacteria causing gonorrhea and syphilis has also become a growing concern, highlighting the potential for untreatable infections to emerge and necessitating ongoing vigilance and adaptation in public health strategies. Emerging and re-emerging infectious diseases pose significant global health challenges. Several factors contribute to increase of these diseases. First, globalization. Increased travel and trade facilitate the rapid spread of pathogens across borders. As populations move, so do infectious agents, making it easier for diseases to emerge in the new regions. Second, environmental changes. Climate changes and urbanization alter habitats, leading to increased human-wildlife interactions. This can expose humans to new pathogens that were previously isolated in animal populations. Third, antimicrobial resistance. The overuse and misuse of antibiotics contribute to the rise of a resistant strain of the bacteria. These complicates treatment options for previously manageable infections, making them more dangerous. Fourth, population growth and urbanization. Rapid urbanization often leads to overcrowded living conditions with poor sanitation. These environments foster the transmission of infectious diseases. Fifth, zoonotic diseases. Many emerging diseases originate from animals. As humans encroach on wildlife habitats, the risk of zoonotic spillover events increases. Last and the sixth one is the socioeconomic factor. Poverty, inadequate healthcare infrastructure, and lack of vaccination access in certain regions contribute to the persistent and resurgence of infectious diseases. Eyes on ethics using GMOs to stop the spread of Zika. In 2016, the Zika virus epidemic was associated with significant rise in birth defects in South and Central America. As the seasons changed in the Northern Hemisphere, health officials anticipated the virus would reach North America, aligning with the breeding period of Aedes aegypti mosquito, the primary vector for the virus. The Aedes aegypti mosquito's habitat extends into the southern United States, serving as a vector for other serious diseases such as dengue and yellow fever. Various mosquito control methods have been proposed, including the use of chemical pesticides, which, while effective, can have detrimental environmental effects. Consequently, some scientists suggest genetically engineering Aedes aegypti to prevent reproduction, although this approach has sparked controversy. One successful pest control method previously utilized the introduction of sterile males. With this approach, male pests are sterilized in laboratories and released into the wild while they mate with females, resulting in no viable offspring. Repeated release led to a decline on the pest population. This method has shown minimal downsides and has been effective against pests like screwworm fly and fruit flies. A more advanced technique involved using recombinant DNA technology to introduce dominant lethal allele into male mosquitoes. These males, reared in the presence of tetracycline, mate with females, but their offspring die as larvae without tetracycline in the environment. This method was successfully tested in Brazil, resulting in a significant reduction in wild Aedes aegypti larvae and dengue cases. In August 2016, the FDA permitted a UK company to test this method in Key West, Florida, pending local regulations. The use of genetically modified organisms, GMOs, for a vector control has both supporters and detractors. Proponents argue that it could potentially eradicate Aedes aegypti, significantly benefiting public health. However, opponents express concern about the risk of a gene escaping into other species and causing unforeseen ecological issues, as well as potential conflicts of interest due to the involvement for profit corporation. Moreover, Aedes aegypti is not the only vector for Zika virus. Aedes albopictus, or the Asian tiger mosquito, is also known vector and is widespread. Other mosquito species may harbor the Zika virus, but their role as vectors remains unclear. Additionally, Zika can be transmitted through sexual contact from mother to child and possibly via blood transfusions, complicating control efforts. The potential risk and uncertainties surrounding open environment experiments with genetically engineered mosquitoes pose significant challenges. Public health officials are faced with critical questions. Can the risk of a Zika epidemic just the ecological implication of modifying mosquitoes? Are existing mosquito control strategies inadequate or harmful enough to warrant exploring unproven alternatives? Microbiology Connection, SARS outbreak. This is the story of the original SARS outbreak of 2002 and not COVID-19. On November 16, 2002, the first case of SARS outbreak was reported in the Guangdong province, China, with the patient showing symptoms similar to influenza, including fever and shortness of breath. The Chinese government's initial reluctance to share information about the epidemic with the World Health Organization and other countries hindered the effective response to the emerging disease. As cases increased, the slow reaction of public health officials contributed to the spread of the SARS both within China and internationally. By April 2003, the Chinese government initiated a comprehensive public health response that included quarantines, medical checkpoints, and extensive cleaning initiatives, ultimately resulting in over 18,000 people being quarantined in Beijing alone. As the outbreak progressed, SARS spread to various countries, leading to its classification as a global pandemic. Initially, the infectious agent was unidentified, leading to the disease being named Severe Acute Respiratory Syndrome, or SARS. Countries implemented travel restriction, particularly to Southeast Asia. As the outbreak escalated, by the conclusion of the epidemic, there were 8,098 reported cases and 774 deaths globally, with China and Hong Kong being the most affected, while Taiwan, Singapore, and Toronto, Canada also experienced significant case numbers. In Canada, the outbreak was introduced in February 2003 by an infected traveler from Hong Kong who subsequently died. By the end of March, Toronto's public health measures included hospital isolation, home quarantine, and stringent hospital protocols. Public health officials actively tracked contacts of infected individuals, leading to 25,000 people being quarantined. Thanks to these prompt and rigorous actions, SARS was effectively controlled in Toronto by June 2003, just 4 months after its introduction. In 2003, the WHO launched a coordinated effort to identify the causative agents of SARS, which was eventually determined to be a coronavirus linked to horseshoe bats. The genome of the SARS virus was sequenced and published by researchers from the CDC and Canada by May 2003, and the further confirmation of the virus's identity was achieved by the researchers in the Netherlands. The last known case of SARS was reported in 2004, marking the end of the outbreak. In the late November and early December, a hospital in the Western Florida experienced a surge in cases resembling acute gastroenteritis. Patients presented with severe vomiting, diarrhea, abdominal pain, and dehydration. Concerned about the rising number of cases, hospital staff discovered that other regional hospitals were also reporting 10 to 20 similar cases daily. Physicians suspected food poisoning was behind the outbreak, which coincided with the Thanksgiving holiday. Over 2 weeks, 254 cases were documented, but the epidemic subsided as quickly as it began by early December. To investigate further, hospitals shared medical records with regional public health office. Laboratory tests confirmed that Salmonella bacteria caused the infections. The affected patients ranged from young children to seniors, with 3 confirmed deaths due to dehydration. All deceased individuals had pre-existing medical conditions and delayed seeking medical attention. Epidemiologists reviewed medical records and conducted interviews with randomly selected group of patients. As Salmonella was identified as the cause, they investigated potential food contamination sources, focusing on the Thanksgiving holiday meals and foods consumed during that time. Analysis of the interview data linked most cases to consumption of a dish called turducken, which consists of a chicken stuffed inside a duck, which is then stuffed inside a turkey. This dish is not typically eaten year-round, explaining the outbreak's timing. Further investigation revealed that the turduckens were pre-stuffed and sold under 2 brand names at a regional grocery chain. Upon identifying the source, Florida's public health officials alerted CDC, leading to an expedited inspection of the processing facility. Inspectors found salmonella contamination due to inadequate cleaning and improper refrigeration practices. Consequently, the plant was shut down for decontamination, and all affected turduckens were recalled, preventing further outbreaks ahead of the holiday season. Links to learning, educators' resources. These are some of the fascinating resources for not only educators but for all. Personally, my favorite one is George Soper's account for Typhoid Mary, published in 1939 in the Bulletin of the New York Academy of Medicine. The last few lines of the article were particularly riveting and as follows. She, Mary Malone, died on November 11, 1938. Her body was hurried away and buried in a grave bought for the purpose at St. Raymond's Cemetery in Bronx. There was no autopsy. The official death certificate states that the diagnosis was terminal bronchopneumonia of 7 days duration, following chronic nephritis and chronic myocarditis of 10 years standing, and the fact that the disease had been typhoid carrier for her 24 years had been contributory. The funeral possessed elements of irony as well as pathos. It was an odd climax to the curious career of this remarkable woman. It was held at the large Roman Catholic Church of St. Luke's in 138th Street, the Bronx. Only 9 persons were present. Of all those in the city employ who had known Mary Malone and seen her come and go for so many years, there was not one who followed her to her grave. Thank you.